All right. Well, I want to thank the organizers for inviting me to this meeting. I'm really sorry that I can't actually be there with all of you. Unfortunately, a family health issue came up that made it impossible for me to travel, but I'm glad to at least be able to tune into the meeting remotely. Um, I feel like a little bit of an outlier at this meeting. I know nothing about the movements of elephants or badgers or fire ants. I'm not actually a mathematician. I'm a cell biologist who has learned a bit about mathematical modeling, and we don't really in my group focus on the mathematical analysis of movement, but we do think a lot about in a cell biological context about how groups of molecules and the movements of molecules and their interactions within cells give rise collectively to the dynamics of cell organization and cell shape change and movement and multicellular tissue morphogenesis. And we really think about that as a multi-scale problem in which we try to empirically with microscopy follow and characterize quantitatively the dynamic behaviors at single molecule levels, and then use those data combined with experiments and mathematical modeling as a way to understand the collective dynamics. And so hopefully, as I go through this talk, you'll find points of correspondence between the things that we do and the things that I think many of you are thinking about. What I want to do is first sort of give you an overview, because I'm sure that many of you are not familiar with the kinds of things we're working on. I want to give you an overview of the kinds of problems that we're working on, the kinds of questions we're interested in addressing, and the experimental tools we're using to generate data on the behaviors of single molecules, and outline some of the ways in which we're analyzing that data. And then if there's time today and tomorrow, I'll give you some specific examples of how we're using those tools to ask specific biological questions. So just to start off with an overview, I'm a cell biologist, but I have some training in mathematical modeling as well. I've been interested, and my group has been interested for a long time, in understanding how early embryonic cells during embryonic development organize their activities in space and time to do the various things they have to do to go from a single cell into a well-formed multicellular organism. And in particular, we've been interested for a long time in understanding how single cells harness the actomyosin cytoskeleton, a collection of polymers and motors and accessory proteins that generate force to deform cells and redistribute their components. How they assemble and organize the actomyosin cytoskeleton, and how they put it to use to do a variety of different jobs, which are exemplified here from on the left, affecting long-range transport that underlies the polarization of single cells, to building structures like the contractile ring, which provides the force to pinch a cell in two, to operating within many individual cells in a multicellular tissue to generate collective dynamics of cell movement and deformation that shape tissues. And we really think about this as a multi-scale problem in which we're trying to go from the behavior, the components of the cytoskeleton, to its collective dynamics. And also, and this is crucial, we're very much interested in not just in the cytoskeleton itself, but in the dynamic coupling of the cytoskeleton to the myriad of proteins that operate in living cells to regulate the assembly and disassembly of the cytoskeleton, and to dictate all sorts of other behaviors in cells. So embryonic cells have a lot to do in a very short period of time, and they do that by dynamically organizing the cytoskeleton and associated proteins to make transient machines, like you see on the top here, this contractile ring, which is used to do a particular job and then disassembled again, or to set up dynamic states of organization across the cell as a whole, as you see on the left here in a cell that's polarizing by segregating factors to one end. And ultimately, those states are underlain by, at the molecular scale, by local processes of assembly, interaction, force generation, and disassembly. The factors that assemble to make the cytoskeleton and to produce force generate forces, which in turn redistribute components of the cytoskeleton and anything that's associated with them. The various regulatory molecules that cells use to control the behavior of the cytoskeleton are physically associated with the cytoskeleton, and so they too get redistributed by the very forces that they regulate. And so at the end of the day, what we're really trying to understand is how these spatial temporal dynamics of cytoskeletal assembly and force generation emerge from the dynamic interplay of assembly, disassembly, local molecular interactions, binding and unbinding, dynamic coupling to transport, and so on. So that's the sort of the underlying goal of a lot of what we're doing. And we do that, much of that in the context of very early events that happen in single cells, the fertilized one cell embryo in nematode worms. So I want to, by the way, just pause to say, as we go through the talk, I would really appreciate it if anybody just interrupts me at any point in time to ask questions if I'm saying things that are unclear, or if I happen to say the one thing that actually piques your interest, please feel free to interrupt. Okay, so I just want to briefly give you the overall context in which we're thinking about these problems, which is the early embryo of the nematode worm C. elegans. So these, during early development in C. elegans, as shown on the right here, you start with a fertilized egg, and this egg segregates factors to two ends to make the two ends different, and then it divides to make two daughters, which have now different states. And those daughter cells typically go on to repeat that same thing. They segregate factors and divide again asymmetrically to produce yet different daughters, and so on. And this is the basic process by which, through a sequence of asymmetric divisions, the early embryo elaborates different cellular states that are going to underlie making the different parts of the embryo. And this is just a movie showing you this process happening over time. And this movie already hides an enormous amount of complexity that happens within individual cells to make all of this happen. So the fundamental process by which a cell executes this asymmetric cell division relies on both segregating to different poles of the same cell a set of factors that regulate the various processes that make those cells different, and then coordinating that with the assembly of the machinery that then divides the cell in two. So I just want to sort of introduce you briefly to some of the main components of that system, because I'm going to be talking about their analysis. So one sort of key component of this system that governs asymmetric cell division is a set of proteins that we refer to as the PAR proteins. The PAR comes from partitioning defective. It's the description of the genetic mutant phenotype caused by mutations in any one of these proteins, where they fail to partition the right factors to two ends of the fertilized cell, so that when it divides, the two daughter cells no longer inherit the right stuff. And the key point of interest about these proteins is that they localize in polarized cells to the cell surface in two different complementary domains. So one set of proteins, which I'll refer to as the anterior pars or the APARs, which comprise at least four proteins whose names don't matter at the moment. I'll introduce them if they're important. And the PPARs, which again are a collection of proteins. The APARs and the PPARs become asymmetrically enriched either on the anterior pole or on the posterior pole. And early genetic studies suggested that the basis for this comes down to mutual antagonism, a kind of competition between these two sets of proteins for occupying the cell membrane, which has essentially an all or none outcome. It's essentially a bistable system in which either the the green proteins win locally or the red proteins win locally. And the outcome of that competition is biased by a cue that's associated with the site of sperm entry. And that cue biases the competition so that red proteins win on one end and the green ones win on the other. Okay. And that may seem abstract, but the underlying basis for this competition, we now know, comes down to, sorry, I realized I forgot to hide one element of that. Permit me a quick, here, let's remove that offending graphic. So the basis for this competition lies in the behavior of the individual components of this system, which are present in the cytoplasm, diffusing around, and can bind to the membrane. And when they're at the membrane, they either diffuse around or they couple to the actomyosin cytoskeleton in ways I'll talk about in a few minutes. And they interact with one another in such a way as to either promote one another's dissociation or, not shown here, to promote one another's recruitment. And it's this system of either mutually antagonistic interactions between red and green proteins or mutually supporting interactions within the groups of green proteins, red proteins, that give rise to these bistable dynamics. Okay, so in the early embryo, the basic events that we think about during polarization can be broken down into two steps, a symmetry breaking or polarity establishment step, and what we refer to as polarity maintenance. So during the polarity establishment step, the green proteins, the par A proteins that will eventually become enriched, start out uniformly everywhere, while the red proteins are in the cytoplasm. The appearance of the sperm cue on one end trigger this rapid redistribution of the green proteins towards the opposite end, which is driven by long-range, active, motor-driven flows along the cell membrane that transport the green proteins to one end and depletes them from the other end. Depleting them from the other end removes their inhibitory influence that allows the red proteins to come up in that domain. And then, once the cue is gone, a short while later, during what we call polarity maintenance phase, these two sets of proteins maintain dynamically stable asymmetries. And they do that through a combination of two things. And I'll tell you more about the details of this later, but they do that through a combination of these mutually antagonistic interactions, and also through persistent asymmetric patterns of flow just beneath the cell membrane, which continues to actively redistribute these polarity proteins. So the thing that makes this system really complex and interesting to study is that the polarity proteins regulate the asymmetric distribution of the cytoskeletal components that drive this flow, and the flow redistributes the polarity proteins. So these two processes are dynamically coupled to one another, and trying to figure out how that coupling works is sort of a central part of what we've tried to do in this system. Okay, so I also want to introduce to you the actomyosin cytoskeleton, which is the main force generator in this system. So in all non-muscle animal cells, the actomyosin cytoskeleton consists of essentially the same proteins that power contraction in your skeletal muscles, for example. But they're organized in a much looser way. So it's basically the cytoskeleton consists of actin filament polymers, which are assembled from individual subunits and are dynamically assembled and disassembled on rapid timescales, as I'll show you in a minute. Various cross-linking proteins that physically link the filaments together into networks or bundles, and complexes that contain lots of individual myosin motors that exert forces on the actin filaments, that try to walk along actin filaments and exert forces that ultimately power contraction. So we actually know a good deal about the machinery that controls the assembly and disassembly of the cytoskeleton. None of the details matter, but what I just want to highlight is that cells have ways of controlling the assembly of different actin architectures, including long unbranched actin filaments that are controlled by proteins called foramins that ride at the growing ends of actin filaments and feed subunits onto their growing ends. And they can also form branch networks using protein complexes that bind to existing filaments and catalyze the formation of a side branch. They can also, through various cross-linking and bundling proteins, set up larger scale networks, either randomly oriented filaments or linear bundles. And they also, cells also have machinery that drives the active severing and depolymerization or disassembly of filaments. So a cell controls dynamically the architecture of actin networks by controlling assembly, cross-linking, and disassembly. And they control forces produced within those networks by controlling the assembly and activation of bipolar myosin filaments that contain many individual motors, each of which is generating force. So many of you are probably familiar with the actomyosin cytoskeleton that operates in skeletal muscle. This is a picture of a single sarcomere in muscle, a single contractile unit, where you have the actin filament polymers that are connected at either side and oriented in such a direction that these big motor complexes called myosin thick filaments have motor heads on either side, which walk towards specific points along the filaments, towards their so-called minus ends. And it's this architecture that promotes walking of motors in a particular direction that generates net contractile force. In an actomyosin cytoskeleton, the filaments and motors are present in a much more disorganized state, and yet they still generate contractile force in ways that we still don't fully understand. So I just want to pause here, just leaving aside the details. I want to give you an intuitive, coarse-grained view of how these networks behave. So what we now understand is that at a sort of coarse-grained level, one can think about a network of filaments and cross-linking proteins and motors as a kind of material. And on a very fast time scale, this material behaves more or less like an elastic material. If you apply a force to it, it will stretch. It will resist that stretch with an elastic restoring force. But if the individual components of the network can turn over rapidly, if the cross-links can bind and unbind, and if the actin filaments themselves can assemble and disassemble rapidly, then the same network, which is stretched, will turn over strained elements and replace those turned over strained elements with unstrained elements. And that will dissipate the elastic force that resists deformation. And so on a longer time scale, turnover endows these networks with effectively fluid-like properties, characterized by an effective viscosity, where in response to an applied force, they will flow on time scales that are much longer than the turnover time of the network elements. And the myosin mini filament sitting within this network at the same time generate internally contractile tension forces that try to contract the network. And so a macroscopic description of a network like this comes down to thinking about how active forces powered by local densities of myosin motors drive the deformations of an effectively viscous fluid. And so I just want to show you, so first I'll just say that in animal cells, such as the eggs we study, the actomyosin cortex is sitting just beneath the plasma membrane in a thin layer, a couple of 100 nanometers thick typically. And it acts within that surface layer to generate contractile forces that are balanced by internal hydrostatic or osmotic pressure forces that resist the compression of the cytoplasm. And so by controlling the local contractility of the cortex, one can either drive cell shape changes, as you see on the left, or by controlling gradients of contractile tension within the plane of the cortex, one can induce local flows of cortex and things associated with it from one part of the cell to another. So here are some examples. This is the one I'm going to talk about more a little bit later, in which long-range cortical flows in C. elegans embryos drive the segregation of polarity proteins. Here's an example on the right in which long-range flows of actin and myosin in the cortex of amoeboid cells engages adhesions on the underlying substratum and basically makes a mechanism to drive the crawling of this amoeboid cell through its environment. The actomyosin cytoskeleton is also used to generate dynamic modes of contractility, such as the pulsatile contractions that you see here, and these are used to generate large-scale tissue deformations. I won't say more about that right now. And the same machinery is also used to generate, on the fly, as it were, the dynamic assemblies like the contractile ring that you see here, which assembles to do the specific job of pinching a cell in two and then disassembles again. Okay, so the actomyosin cytoskeleton is this versatile collection of motors and filaments and accessory proteins that generates macroscopic dynamics of deformation and flow. And we have been using the early C. elegans embryo as a model system to think about this problem, because in the early C. elegans embryo, the same cell over the span of about 10 or 15 minutes uses and remodels the same machinery to do variants of all of those behaviors I described. So during the initial symmetry breaking and polarization steps, the cell makes long-range flows that segregate polarity proteins. Less than 5 minutes later, it uses exactly the same machinery to weave a contractile ring. Shortly thereafter, in the two-cell stage embryo, it manifests this mode of pulsatile contractility. So these cells can dynamically remodel the actin cytoskeleton to do a whole range of different jobs at different times for different purposes. And a big part of what we're trying to understand is how the microscopic how control of the microscopic dynamics of assembly and disassembly and force generating interactions gives rise to these macroscopic behaviors. Okay, so as I said at the beginning, what we're really interested in here is the problem of what I would call mechanochemical pattern formation. So we're interested in understanding how large-scale spatiotemporal dynamics of polarization or cell division emerge from this dynamic coupling of actomyosin networks and the forces they generate and the deformations they create, and biochemical networks, exemplified here by the paraproteins, that themselves embody systems of reaction and diffusion interactions that can give rise to spatial patterns. But what we're particularly interested in is the dynamic coupling between these, how the forces and flows generated by the networks transport components of the biochemical networks, how the biochemical networks in turn regulate specific features of assembly or disassembly or motor activity, and how macroscopic dynamic behavior emerges from that dynamic coupling. So one can think about these problems from a modeling perspective by coarse graining everything and just sort of thinking at a continuum level about the macroscopic behaviors of the cortex as a material and the macroscopic behaviors of large ensembles of molecules. So for example, we can capture the dynamic behavior of an active viscous fluid that I described a little bit earlier by just writing down equations that describe how the local tension or stress within the cortex is governed by a combination of active stresses produced by the actomyosin cytoskeleton, which depend on its density and other things, and passive stresses, which depend on the local deformation rate and some effective viscosity. And one can write down a force balance equation that balances the gradient of this active stress, which has units of force, against the frictional resistance proportional to velocity of the movement of components of the network. So that gives you a continuum level description of active dynamic flows in a cortical cytoskeleton. And at the same time, one can write down standard reaction, advection, diffusion equations that describe how local densities of elements at the membrane change through local binding and unbinding and so on, transport by flows and diffusive processes. And one can couple these together by making the various components in the system depend on local densities and so on. So I think probably this level of description is most likely familiar to many of you. And it's a very useful way to think about these problems. But for us, especially as experimentalists, the meat of this is to really understand how the dynamic coupling that's embodied in mathematical terms in this equation actually works. And that is, how do individual components of the reaction diffusion system couple dynamically to flow? What are the actual interactions among components of the system that govern spatial pattern formation? How do the asymmetries emerge through some combination of local binding and unbinding and transport? And how do those individual properties, binding, unbinding, and transport, depend on specific interactions among different components in the system? And so for us, the way to meet that challenge has been to develop a set of tools that we can use in living cells to monitor the dynamic behaviors of individual molecules as a way to get direct access in exactly the same way that we heard a minute ago when we get by putting a GPS tag on an elephant. But here, by putting a fluorescent tag on individual molecules in a way that can be genetically encoded so that you can make cells that just make proteins that are fluorescently tagged, and then engineer the cells in such a way that they're expressing sufficiently low levels of those molecules that one can, or in which a sufficiently low fraction of molecules has been labeled fluorescently, so that one can follow the individual molecules around and observe what they're doing and infer their dynamics. And so the general approach which we've used to do this, which is completely general in this organism, is to use genetic tricks to make worms that make a protein of interest which is fluorescently tagged. In these embryos, they're produced by mothers that install into the newly fertilized eggs all of the protein that they need to get through early development. And one can use genetic tricks to induce in the mother, before she lays these eggs, a depletion specifically of the proteins that are labeled fluorescently. And what that allows us to do is it allows us to go from an image that looks like this, where all of the molecules made by the mother are fluorescently tagged. This happens to be a protein that labels the actin cytoskeleton, so you see a filamentous network at the surface of the cell. But we can tune the expression of this protein experimentally down to very low levels, so and then use a technique called turf microscopy or near turf microscopy that allows us to selectively illuminate just a thin layer just at the very surface of the embryo, which has been pressed against a glass cover slip. That allows us to excite fluorescence and record fluorescence only from the subset of molecules that are sitting right at the cell surface. And that allows us to observe those molecules with high signal to noise. So we can go from something where you have full density down to images that look like this on the bottom left, where now the same probe is expressed at very low densities, such that we can see individual speckles. And we can confirm by measuring the fluorescence intensity of those speckles that those intensities are Gaussian distributed, meaning that they correspond to the fluorescence coming from a single molecule, a single fluorescent molecule. And then one can film those molecules at frame rates up to 30 frames a second to get data that looks like this, where one can now see individual speckles that appear and then move locally and then disappear again. This happens to be, in this case, the subunit of actin filament, of the cytoskeletal polymers we care about, which is fluorescently tagged. And so here, when these speckles appear, we interpret that to mean that an actin subunit has added onto the end of a growing filament. The speckle becomes visible because in the cytoplasm, a free monomer is diffusing so fast that the image of that molecule on the camera is blurred out during an acquisition period of 30 milliseconds. Whereas once the molecule binds an actin filament, its mobility goes way down, and so now we can see it as a bright speckle. So the appearance of a speckle signals an assembly event. The disappearance of a speckle signifies a disassembly event or photobleaching. And I'll come back to that later. And during the period of observation, the movements of that speckle inform on local movements of whatever protein has been tagged. Here's another example. This is now one of the anterior PAR proteins, which, as you can see, even when reduced to single molecule expression levels, is highly enriched on one end of the cell. And you can again see individual molecules appearing, moving around, and disappearing. And I think if you stare at this movie for a while, you'll also see that there appear to be different modes of mobility. There are some molecules that appear to be moving quite slowly and others that appear to be moving quite fast. So here again, these kinds of data give us direct access to binding events where a molecule appears at the membrane and becomes visible. It gives access to mobility, and it gives access to unbinding events when a single molecule disappears again. And even more importantly for us, this is a technique that we can combine with experimental manipulations where we manipulate one molecule which we think might control some aspect of the observed molecule's behaviors and see how those manipulations change those behaviors. So this becomes a very powerful experimental tool for us. Here's just one more example, which I'll sort of come back to a bit later, of observations, in this case, of direct observations of actin filament assembly, where we have now labeled the molecule that sits at the growing ends of actin filaments and feeds subunits directly onto its growing end and remains associated with the growing end, so that movements, which you can see if you look at this movie for a while, directed ballistic movements, give a direct readout of local assembly of individual actin filaments within this system. And here's just to show you that if one does some image processing, which I'll tell you more about later perhaps, you can actually track these individual molecules and get a direct quantitative readout of local rates and direction of assembly. Okay, so those are the kinds of data that we can generate in this system. Data, single molecule data, which reflects different modes of movement from binding and unbinding between cytoplasm and the membrane, local movements within the membrane, local movements associated with filament assembly. And then for us, a key challenge has been to extract from that kind of raw data trajectories of individual molecules or particles, which we can then interpret to generate quantitative measurements. And I just want to outline the approach by which we do this. I just want to check the time here. Okay, good. Excuse me. I just want to see how we're doing for time. So the typical approach to analyzing these data goes through two steps. One has to first, from raw data, detect lots of individual bright speckles, and then one has to link those speckles together from frame to frame in order to compile trajectories. So the detection step is relatively straightforward. One starts out with raw data like this. Here you just see a blow up of a small region of the image on the left. And one uses a simple bandpass filter to filter out both low spatial frequency and high spatial frequency information. And that gives rise to now data that looks like this, where one can go through and to systematically detect local peaks in fluorescence intensity within the image. And that addresses all of the individual peaks within an image like this. And then for each one of those peaks, one can go back and look at the raw data. And from the distribution of fluorescence intensities around that initial peak, one can then find the center of mass of the fluorescence intensity and assign an actual position to the particle. And one can use a threshold on the background subtracted fluorescence intensity to pick out only those particles above a certain brightness that represent molecules, as opposed to things that just represent local statistical noise in the image. And that gets you to something that looks like this in an individual frame, where one has both detected individual molecules and determined their localization to an accuracy of about 50 nanometers. Okay. And then, of course, the next step is one wants to link those molecules together to form trajectories from frame to frame. This is a relatively straightforward process if the density of molecules is low enough that the mean displacement of a molecule from one time point to the next is significantly lower than the average distance between molecules, because then you just use simple minimization strategies to assign pairs of molecules. But very often we want to collect data that's at a higher density, because as you sort of saw in the last talk, the number of individual trajectories that you can analyze gives you the statistical power you need ultimately to be able to compute behaviors in the system. And so we're always trying to get to higher densities. And when you get to higher densities, you run into tracking problems, because now there's the possibility that a molecule that you see in one time frame could become any one of several other molecules in the next time frame. And there's the possibility that two molecules will move together to occupy the same diffraction limited spot, which shows up as a kind of a merger event, or they can separate from one another. And so a more complicated method to do this, which is what we typically use, is to divide the problem up into two steps. This is using software and tools developed by Kahlua Jacob, and this is a great paper if you're interested in these methods. So the idea is to first go through and just link individual particles frame to frame, and then subsequently to take these trajectories and link the trajectories together to detect several different kinds of possible higher order events, either to detect gaps in a trajectory where a molecule may have failed to be detected for a few frames, to stitch together trajectories into longer ones, or to detect these kinds of merge and split events. The basic approach to doing this, which I won't go through the details of, amounts to solving a linear assignment problem, where the idea is to assign probabilities for these various different linking events and costs associated with those probabilities, and then to do at each frame in time a global optimization within that frame to determine the most probable linkages, and then to follow that over time. So that approach, what that approach gets you to in the end, is it gets you to a list of trajectories for individual molecules that gives you their positions over time, and those trajectories, the length of those trajectories depends on the behavior of the molecules and the accuracy of your tracking, and so on. Now I want to just highlight some of the ways in which we are analyzing this kind of data. The first, analyzing particle motion. This is probably for most of you sort of trivial, but this is one simple way that we often analyze the behavior of different molecular species is just by plotting the mean square displacement versus lag time tau, and plot it on a log plot. This gives you a very simple way to distinguish different classes of motion. So plots with a slope of 1 signify Brownian diffusion, plots with a slope of 2 signify ballistic motion. Plots with slopes less than 1 signify some form of anomalous or restricted motion or anomalous diffusion. And of course, real data never looks quite this clean, but we can use this as a sort of a reference to do slightly more sophisticated analyses. So let me give you an example of the kind of analysis that one can do using this approach. So one thing that we would like to be able to do from data like this is to infer different different classes of motion for the same molecular species that might indicate different underlying states for that species. So here's an example. This is completely analogous to the problem you heard about a few minutes ago in the last talk, where you're trying to look at the motion of elephants and infer something about the state of the elephant. So in this case, the analogous situation is that we're interested in molecules that could exist in different binding states and different complexes with other molecules, which might have different mobilities associated with them. And the question is, can we distinguish those different mobility states and then track molecules as they walk through them? And so in this example, we're looking at a protein which is one of these so-called anterior PAR proteins on the membrane. And we know from a lot of biochemical studies that we and others have done that the molecule we were looking at, a molecule called PAR6, can exist in a complex, in one of two different complexes, one of which is scaffolded by an oligomeric protein called PAR3, and the other of which is scaffolded by another molecule called CDC42. Just think of them as blue and cyan complexes. But we know from looking at the behavior of PAR3, the scaffold in living cells, that PAR3, it's an oligomeric protein that forms largish clusters whose mobility is restricted through interaction with the cytoskeleton. So it exhibits slow subdiffusive motility. If we look at, on the other hand, if we look at PAR6, we can see that a subset of the PAR6 fluorescence is also associated in clusters. And we know that these clusters coincide with the clusters of PAR3. So we know that some of the PAR6 is in clusters containing PAR3. So it's in this complex. But there's also this diffuse pool of PAR6, which is not associated with PAR3 clusters, which we assume must represent PAR6 in a second complex associated with CDC42. Now, looking at this level of total fluorescence intensity, it's very difficult to go any further. But by using single molecule approaches, we can actually now follow the trajectories of all of the individual PAR6 molecules, and we can plot mean square displacement as a function of tau. And what we now see is that these trajectories exhibit a large range of different, a spectrum of behaviors. But there's one population which has slopes alpha, lying around 1, indicating Brownian motion, and then another family of plots, which I've highlighted in blue here, for which the alpha is much less than 1, which we assume represents subdiffusive motion. One can go further and fit these data to a simple anomalous diffusion equation to extract both an anomalous exponent alpha and an effective short-term diffusivity D, and plot as a scatter plot for these data, those two different quantities, molecule for molecule. And now you get a scatter plot that looks like this, where you can detect one subpopulation highlighted in green here, which is effectively diffusive and which has high diffusivity, and a second subpopulation, which is indicated by the blue here, which has very low short-term diffusivity and has and is subdiffusive. And we can use, in fact, simulated data for Brownian diffusion, and perform exactly the same analysis to see that this distribution that you see for the orange molecules corresponds to the distribution you would expect to see for simulated Brownian diffusion at about 0.2 micrometers squared per second or so. Okay, so that suggests that there might in fact be two distinct mobility states. And of course, we would like to map those two mobility states onto these two different complexes. How can we do that? Well, we could do the same exact kind of plot using by imaging and tracking PAR3 clusters. So that's what this distribution looks like. And then we can overlay on top of that the distribution for all of the PAR6 molecules. And what you see is that there's some correspondence, but there's a large fraction of single molecules that are up in this region that indicate Brownian diffusion, which we don't see for PAR3. And we can now engineer a cell in which the par6 molecule has been modified so that it can no longer bind to this protein CDC42, so it can no longer enter this complex. And now what we see is that crippled par6 molecule, which can now only bind to par3, has a distribution of diffusivities in tau that lay almost directly onto the par3. So this tells us quite clearly that there are two different mobility states for this par6 molecule that we can detect. One of them corresponds to par6 bound in a complex with par3. The other corresponds to par6 bound in a complex with CDC42, which is highly diffusive. And moreover, we recover from this kind of analysis a quantitative data set, which tells us everything we need to know about the characteristics of motion associated with those two states. So you could imagine how this kind of data could be used to tightly constrain the kind of hidden Markov models that you heard about a little while ago. Okay, so I want to just say and give you one more example of another class of measurements that are very important for us, and that is to measure local binding or assembly kinetics. So one can, by looking at data like this, what one is essentially seeing is one is seeing individual events where a molecule that was in the cytoplasm comes up to the membrane, moves around for a while, and disappears again. And so in principle, by observing appearance and disappearance events of single molecules at the membrane, one should be able to infer the kinetics for this binding and unbinding. And similarly, if one is looking at the subunits of an actin filament polymer, then the appearance represents assembly events and the disappearance represents disassembly events. So we would like to recover those kinetics. How can we do that? Well, a simple way to do that is to collect an ensemble of trajectories from any one of these kinds of data, and then plot so-called release curves, where you plot as a function of time the number of the total number of tracks that have a residence time longer than a given time. So if molecules are disassembling or unbinding with first order kinetics, then these release curves should be exponential or linear on a log plot over some time interval. And that's exactly what we see for many of these kinds of data. One can then read directly off of the slope of this curve an effective disappearance rate, which is the sum of the true dissociation or unbinding or disassembly rate and a rate which characterizes the rate at which single molecule fluorescent molecules are photobleached and become invisible. So one needs to decouple those two measurements from one another. And the approach that we use to do that is to run a series of experiments where we vary the duty ratio of laser exposure. So we systematically control, leaving everything else constant, the amount of laser light that the molecules are exposed to, and so the photobleaching rate. And one can then obtain a sequence of such curves, one for each duty ratio, and extract the observed loss rate for each of those duty ratios, and then fit that to an equation that describes the total loss rate as a sum of a disassembly rate and a photobleaching rate, to pull out a true dissociation or disassembly rate and a photobleaching rate. So that gives you directly a quantitative measurement of disassembly or dissociation. Now, so I'm going to I just want to summarize here. So that gives one ideally a way in which one can measure, local dissociation or disassembly rates. But as I showed you a few minutes ago, for many of the molecules that we're looking at, there are a heterogeneous population of single molecules at the membrane, different species appearing in different complexes. And so one might expect that the data is not always going to look as clean as this. That is, that there may be multiple dissociation rates associated with different mobility states. So one of the things that we're now very interested in doing, which I would love to talk to many of you here about, especially if I were there in person, is how one can begin to leverage, for example, the kinds of hidden Markov modeling approaches we just heard about as a way to extract sort of measurements of dissociation rates or disassembly rates associated with different species that happen to be in different states by combining information about states that can be inferred from mobilities and information about trajectory lengths that can be inferred from their lifetimes. So I want to now, I think I'm going to I want to stop there because I would like to actually, I've been speaking into the void for the last 50 minutes. I want to stop and take questions from people, and I will then, in tomorrow's lecture, give you some examples of how we're using these approaches to actually ask some specific biological questions. So I will stop there and take questions. Yeah. So what we're typically doing here is what the raw data looks like is a list of trajectories of varying lengths. The lengths depend on both the residence time of the molecule that you're looking at the membrane, and is also to some extent limited by particle tracking errors, which we've learned how to minimize. And so typically what we're doing is we are, for any particular lag time tau, we are just going through all of the individual trajectories and extracting as many independent measurements of lag time for that tau as we can. So for a trajectory of length 3 tau, we can get 3 measurements of lag time versus tau from that trajectory. And that means that the number of measurements of mean squared displacement for a given lag time will be high for short lag times, and it will fall off for longer lag times. And what we typically do is just set a cutoff where we're only going to report a mean square displacement for when we have more than some number of observations. In practice, we typically set that cutoff at 100 observations, and one can just see in the MSD plots when the MSD predictions start to get poor because the variance in the individual measurements goes way up. Did that answer your question? So there's no renormalization, but we are keeping track of the statistical power of the individual measurements. Yes, that's absolutely, that's right. So we're only computing measurements on observed data. And yeah, sure. Yeah. 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 Okay. That's so let me go back to that. So that's the plot on the bottom left here, right? And so this is a so if you are fitting, this simple equation MSV equals 4D tau to the alpha to any data, the accuracy of the fits, there's error in the fits, of course, and that error depends on the length of the trajectory, on the number of observations you have along a trajectory. And so this is there's a sort of a beautiful classical paper by Hong Chen that sort of goes into this in detail that I could send you a reference to if you like. But the idea is that when you do these fits, you don't predict that you're going to recover the exact value of either alpha or the diffusivity. You're going to recover a scatter plot that looks something like what you see at the bottom right here. So in fact, what we did here was we starting with the observed data, we said, okay, in the observed data, we have a certain number of trajectories with a certain length distribution. And what we want to now do is create simulated data under the assumption that molecules are just undergoing Brownian diffusion with a single diffusivity, which we draw, and so we draw displacements just from a Gaussian distribution at each time point. And we resimulate exactly the same number of trajectories with the same length distribution with that single assumption. And then we subject those data. We also actually add in the measurement error, and then to produce a simulation of measured trajectories. And then we do exactly the same fitting procedure. And so what you see there, that white scatter plot, is the predicted scatter plot distribution of diffusivities in alpha that you would expect to see for a population of, for data that comes from an underlying pure Brownian diffusion process. And so for us, this was just a way to say this scatter plot distribution that has a characteristic diagonal and has a distribution of alphas and diffusivities looks exactly like what you would expect if what you're measuring is data from an underlying population of molecules that are just undergoing Brownian diffusion. That's exactly right. The tilt just comes from the error inherent in fitting an equation to a limited number of data points. It's just that, yeah. So this gives us a way both to verify that this is what you would expect for pure diffusion, and also to get an estimate of what would the diffusivity have to be in order to reproduce at least approximately the scatter plot distribution of points. Yeah. Yes. Yes. Yeah. Yes. Yeah. 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 Yeah, no, it absolutely makes sense. It's a question of different behaviors on different time scales. And it's actually so here we're faced with an inherent limitation experimentally, which is that in order to obtain accurate tracking of rapidly diffusing molecules, we have to use short duration exposures to get time resolved information, and we have to use a lot of laser power to get high signal to noise. And when we do that, it means that we're pretty rapidly photobleaching molecules. So that limits the duration of the observation of a single molecule to about a second. You'll see that the cutoff here is at less than 1 second. So we do indeed, if we either use tricks to increase to use molecules that are labeled with more photostable fluorophores, or we're looking at things that have lower mobilities overall, so that we can get accurate tracking with lower signal to noise and use less laser power. We can extend these observations to longer times. And when we do that, what we typically see is that the MSD versus tau plots have much more interesting shapes. So a plot that might be linear on this time scale I'll actually show you an example tomorrow of, of a molecule that sits at the cortex in clusters, which has behavior on very fast time scales that looks diffusive. It then becomes subdiffusive at intermediate time scales. And then it becomes what looks like diffusive again at very long time scales. And those are time scales on which the turnover of the cortex makes the cortex as a whole start to behave like a fluid again. So these clusters behave like something that's effectively diffusing. So this approach can provide incredibly rich data, and what we've spent a lot in the last 10 years learning how to do is both how to from the theory to think about what does the optimal data look like to answer particular kinds of questions, and then from the experimental side, how do you use various experimental tricks to optimize the data acquisition. And I would say that the place that we're still lacking in as much sophistication as I would like is exactly thinking about the kinds of things that you all are discussing at this meeting, which is how do you sort of think mathematically about the dynamics of ensembles of trajectories like these. Yes. Yeah. So no, this is a very good question. So there is two sort of forms of active transport that we observe in the system, depending upon spatial and temporal scales. So on a relatively fast time scale, the local forces generated by individual motors are generating transient local movements or displacements of the cytoskeleton, things associated with the cytoskeleton, and the motors themselves. And that looks like, on an intermediate time scale, that almost looks like diffusion. It's not diffusion, but it looks, sort of if you just look at the distribution of displacements, it looks a lot like Brownian diffusion. Sometimes you can see, if you look at a Van Hoff distribution, you can see that the tails are not Gaussian. But then there's also on a longer time scale, there are these sort of coherent long range flows that I showed you examples of. And so it turns out that for if you're looking at molecules that are diffusing at the membrane, they in fact sense that flow, but the diffusivity is high enough relative to the flow that it's very difficult to detect just by looking at MSD plots like this, to detect the signature of the flow. Because you can't, there is some time scale on which on which transport will dominate diffusion, but that time scale is much longer than the time scale on which we can observe molecules in most cases. So it's there and we can detect it, but it doesn't affect these observations much. Yeah. Okay. 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 Thanks, everybody.